Hi friends, welcome to STUDYHIGH.Pk and subscribe, keep in contact for more videos and update. Today we are going to start our next chapter lipids after completing our discussion on chapter carbohydrates, medical biochemistry lecture 1 of chapter lipid. The basic lecture, what are the lipids and its classification into different types that we will discuss today. So let's start our discussion on this lecture. Medical biochemistry chapter lipids. First of all, I want to inform you what are the lipids. The simplest definition of lipids is that lipids are the heterogeneous compounds that consist of fatty acids and alcohol, but they can also consist of different type of components other than fatty acid and alcohol that we will discuss in classification of lipids. But first of all, I want to discuss the common property of all type of lipids or the diversity of lipids in human body. Almost just assume this is a non-polar solvent in a container. It consists of benzene. Benzene is a non-polar hydrocarbon ring or chloroform, whatever. So this is a non-polar solution. If you take any lipid, maybe phospholipid, any lipid, this is a lipid, and you enter or dissolve it in non-polar solvent. Almost all the lipids are dissolved in this aqueous non-polar solution. So lipids will not dissolve in water or aqueous solution, but they will dissolve in non-polar solvent like benzene, hydrocarbons. So this is the common property of all type of lipids. That's why sometimes we define or scientists define lipids in that way that any molecule that is soluble in non-polar solvent and has a biomolecule origin, or any organic molecule that has a function in human body but and also dissolve in non-polar solvent is called lipid, consists of fatty acid and alcohol as their major component. So almost all lipids are dissolved in non-polar solvent, but some lipids like phospholipid, they also consist of a hydrophilic head because of phosphate bond, so fatty acid tails and hydrophobic head. So this is their phosphate group head that is hydrophilic, whereas this is a non-polar tails of this phospholipid that are hydrophobic and could not be dissolved by water or in aqueous solution. So if such type of lipids, mostly these lipids involve in formation of cell membranes. That's why in cell membranes, the hydrophobic tail are in towards the center, whereas the hydrophilic head is towards the aqueous environment of extracellular matrix. Similarly, in treating cystic fibrosis or different type of diseases, scientists form liposomes. We will discuss in our coming lectures what are liposomes, their effects and how they form. So liposomes in their original form, they have different type of arrangement, consist of both hydrophobic and hydrophilic region. So liposome are formed. So if you dissolve this disordered lipid constituents into a lipid or aqueous solution, what will happen? If this is an extracellular matrix aqueous solution, all the heads that are hydrophilic will be projected towards the water or aqueous solution, whereas hydrophobic tails will be projected toward the center. In this way, they form a covering the aggregation called liposome, different functions that we will discuss. So no other functions. First important function of lipids is that they involve in signaling hormones. Some lipids like phosphatidylinositol, when these all cell membrane of cell almost consist of receptors, when some hormone, some any type of antigen antibody bind with this receptor, there are some hormones are produced or released from the cell membrane that can trigger the action or different type of action by reacting with the receptors present on the nucleus and can cause a different type of cascade of reactions in the cell. So these hormones that can cause or can act as a messenger, like phosphatidylinositol can also act as a second messenger system. So this is also a function of phospholipids or simply lipids. lipids has also different type of functions. Like sphingomyelin is also a lipid that is present in nerve fibers. You must know nerve fibers sometimes consist of a material that is called sphingomyelin that is non-polar, insulated. It does not conduct impulses, but because of presence of this sphingomyelin, the nerve impulse that is coming from one side jump from this part containing sphingomyelin to the next. As a result, the speed of nerve impulse increases to a large extent. So they also have a role in conduction of impulses. Different type of lipids, like if this is a person, this is you, and you on a marriage of your cousin or some relative, you eat a lot of meat or lipid containing food. As a result, what will happen? Now your body has a large amount of fatty acid lipids. Then what will happen? The excess fatty acids will be stored in your adipose tissues in the form of triglycerides. When when you again feel hungry after some time, after 2 to 3 days, when you found no food to eat and medicine has ended, then what will happen? This adipose tissue that has stored your extra fat, extra triglycerides in the form of triglyceride, it will start hydrolysis and will be broken down into original fat and alcohol. Fat and alcohol that involve in formation of lipids like triglycerides and alcohol glycerol. And this fatty acid will pass to the mitochondria, and mitochondria will process it through citric acid cycle, electron transport chain, and will generate ATP so that you can live or use this ATP for performing different type of functions for thinking etc. So this is the basic process you and use of different uses of lipid in our body, in our hormone system, receptor signaling as second messenger, in nerve impulses, in storage and producing ATP, so etc. Now we are going to discuss the different type of lipids. Lipids, we classify lipids into three important types, like simple lipids, complex lipids, and derived lipids. And what is the difference between these types? First of all, you must know how lipids are formed. Just assume this is a fatty acid. Fatty acids and alcohol are the two major components of lipid. They react with each other and through our form a bond. Ester bond is present between the fatty acid and alcohol. So this is the fatty acid and this is alcohol that react with each other and form a ester bond with each other and involve in formation of lipids. So fatty acid and the alcohol are the two important components of all types of lipids, whether simple, whether complex, and they combine with each other with the removal of H2O and form a ester linkage between its carbon atoms, C double bond O and another O, and here is a carbon of alcohol. No, these fatty acids and alcohol are the basic components that involve in formation of lipids. Now, what is simple lipid? If any lipid that consists solely of fatty acid and alcohol and does not contain any other substance other than fatty acid and alcohol, then we will call it is a simple lipid. As it consists of only fatty acid and alcohol, we further classify simple lipid into different types on the basis of nature of alcohol, whether it is monohydric or dihydric or polyhydric, or on the basis of fatty acid, whether it is unsaturated or saturated fatty acid. So these are the different types of classification of simple lipid. So we are now going to classify simple lipid into different types. The first type is fat, second type is oils, and the third type of simple lipid is waxes. So we classify simple lipid into different types on the basis of configuration, whether monohydric alcohol involved in formation of lipid or whether saturated or unsaturated fatty acid involved in formation of lipids. So we classify simple lipids into three types: fat, oil and waxes. And the alcohol that is present in fats and oils is glycerol. It consists of three carbons connected with three hydroxyl groups that can form ester bonds with three fatty acids. R1, one fatty acid, R2, second fatty acid can react with carbon 2 hydroxyl through ester linkage, and R3. So, glycerol is present in fats and oils, whereas monohydric alcohol is present in waxes that consist of a large number of carbons. In waxes, alcohol consists of a high molecular weight carbon and at the end only one hydroxyl group that involve in formation of bond with fatty acid to yield lipids. In simple lipids, I have explained you that simple lipids consist solely of fatty acids and alcohol, are lipids which on hydrolysis yield only fatty acid and alcohol are included in simple lipids, whereas in complex lipids, one, the basic component is fatty acid, and maybe we further classify complex lipids into different types. In some types, cetibonic acid is present as a component of fatty acid. In some cases, oleic acid, palmitic acid, stearic acid. So, further classification is made on the basis of nature of fatty acid. And the second important component that is also a component of all type of lipids is alcohol, and it can be glycerol, it can be monohydric, dihydric, and in some cases, in most cases, it is also sphingosine. Sphingosine is also alcohol that involved in different types of formation of different types of lipids, and these two are also the components of simple lipid. But in complex lipid, there is also a third component that is other than fatty acid and alcohol. That may be a phosphate group, a carbohydrate, a sulfate group, any group. So, in complex lipids, in addition to fatty acid and alcohol, the lipid also contains some other component like fatty acid, like phosphate group, carbohydrate, etc. Like simple lipids, complex lipid also has different types that now we are going to discuss, like glycerophospholipids, sphingospholipids, glycolipids, and how you can differentiate between these different types. You will be able to differentiate after my discussion on these types related to complex lipids. So first of all, complex lipid consists of any component, phosphate group, sulfate group, carbohydrate, any component that is other than fatty acid and alcohol. In addition to fatty acid and alcohol, we can also define complex lipid in another other way. Those lipids who on hydrolysis yield fatty acid, alcohol, and an other component in addition to fatty acid and alcohol that is not fatty acid and alcohol. That may be fatty phosphate or carbohydrate sulfate group. So now we are going to discuss the different types of complex lipids. First of all, we will study glycerophospholipids. And what are the constituents of glycerophospholipids? Their detailed structure we will study in our coming lectures. First of all, glycerophospholipid first of all consist of fatty acid, first component. Second component that is also the basic component of all type of lipids is alcohol. But the alcohol in case of glycerophospholipid is glycerol, whose structure I have explained you in my discussion of previous simple lipid. It consists of three carbons that are connected with three hydroxyl, and when it combine with three fatty acids, then we will obtain glycerophospholipids through ester linkage. So, in glycerophospholipid, one component that involved in formation of glycerophospholipid is fatty acid, and the second component is alcohol, that is glycerol, and third component is phosphate group. And the phosphate group that involved in formation of glycerophospholipid may further be modified with different type of bases like choline or inositol, different type of serine residues, ethanol amine. So phosphate is further connected or modified with different type of nitrogen bases or compounds. As a result, glycerophospholipid also has different types. like lecithin that consists of fatty acid, glycerol, phosphate, and in lecithin phosphate is further modified with choline, a base. Likewise, phosphatidylethanol is also glycerophospholipid. So its detail are different types. We will study in our coming lecture. So first of all, you must know that what are the constituents that involve in formation of glycerophospholipids. Now we will study what are sphingophospholipids and what are the constituents that involve in formation of sphingophospholipids. The first basic constituent is fatty acid, that is also constituent of all types of lipids. Second component is alcohol, but the alcohol that involve in formation of sphingophospholipid is not glycerol, but it is sphingosine. Sphingosine structure I want to show you, it consists of an alcohol hydroxyl group. This is not the exact structure, but I just want to show you how sphingosine and what is the structure of sphingosine look like. And it also consists of an amino group. So in sphingophospholipid, sphingosine is the alcohol, whereas in glycerophospholipid, glycerol involved in formation of lipid. Here sphingosine. And the third component of sphingophospholipid is again phosphate, and it also sometimes further modified with different type of bases, like in sphingomyelin that is present in nerve fibers. Phosphate is further attached with choline base. So in choline consists of almost 3 further nitrogen. We will study its structure in our coming lectures, and 3 further methyl groups. So structure of choline look like, and N nitrogen has a positive charge. It is further connected with another carbon and then with the phosphate group. So in sphingophospholipid, sphingosine is the alcohol that involved in its formation, and one is a fatty acid, and third is a phosphate group that may further be connected with different type of nitrogen bases like choline or inositol, myoinositol. We will study in our coming lectures. Now we are going to discuss a third type of complex lipid, glycolipids. What is difference between glycolipids and glycerophospholipid or sphingophospholipid? Glycolipid consists of one fatty acid, one alcohol. In most of its compound, the alcohol that involved in formation of glycolipid is sphingosine, that is also present in sphingophospholipid. So the alcohol in most cases in glycolipid is sphingosine, whereas the third component of glycolipid is not phosphate, but it is carbohydrate that may be glucose or galactose. So sphingoglycolipids consist of a sphingosine tail and other fatty acid. Here is a fatty acid that consists double bond O, and a third component is glucose or galactose. So this is a structure of glycolipid. In this topic or in this lecture, you must only know that what are the components of glycolipid and details its structure, its functions of glycolipids in human body. We will discuss in our coming lectures in lecture 6 approximately. So the constituents that involve in formation of glycolipids are fatty acid, alcohol that is sphingosine, and carbohydrate that may be glucose or galactose. These are the basic components of glycolipids. The third type of lipids in which we classify lipids are derived lipids that consist of a number of products like steroids, aldehydes, ketones, different type of components. In addition to fatty acid and alcohol, vitamin D that it can be produced by the derivation or breakdown of a bond in cholesterol. Like here is the cholesterol rings, 3 rings for 3 hexagonal rings, 1 cholesterol consists. We will study cholesterol in our lecture 8 inshallah, and it also consists of a 1,5 carbon. So on exposure to light, this cholesterol can be converted into vitamin D. So derived lipids are also present in human body, and we also derive or classify lipids into derived lipids that on further hydrolysis yield different type of compounds. and steroids, aldehydes, ketones, vitamins. All these are also included in derived lipids or lipids who on hydrolysis yield these components, different type of components other than simple fatty acid, alcohol, phosphate and carbohydrate. All are included into derived lipids. So sometimes glycerophospholipids. Now I am going to explain another simple concept. Glycerophospholipid cholesterol, whereas cholesterol ester. We will also we have a complete lecture on structure function of cholesterol. All these are also called neutral lipids as there all the alcohol form bond with all the three fatty acids and they are uncharged. So uncharged phospholipids are also called neutral lipids. So neutral lipids, you must also know what are the three types of neutral lipids important. One is triglyceride, triglyceride, cholesterol and cholesterol ester. All these are also included into neutral lipids. They are uncharged fatty acids, uncharged lipids. So this is the our today's discussion on classification of lipids. In next lecture, very important, we will discuss the number and naming of fatty acids and different types of fatty acids like arachidonic acid that also involve in formation of different type of leukotrienes, prostaglandins, and we will also discuss about structure of different type of hormones like estrogens, progesterones, and testosterone, androgen, etc. Hope you best of luck. Like and subscribe to keep in contact with STUDYHIGH.Pk. Hope you best of luck. Allah Hafiz.